Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Diversity of Plants Zone. In this channel, you will get every necessary information related to the plants and their diversity throughout the world. Before starting my today's topic, I request you all to please subscribe my YouTube channel. My today's video is about differences and comparison of two very important domains, which are Domain Archaea and Domain Eubacteria. I will elaborate their differences and comparison with the help of the comparison chart. So let's start the video. I hope you will enjoy this video and appreciate my work as well. First of all, I will discuss about the Domain Archaea. So, what is Archaea? Archaea are single-celled and simple microorganisms that live and thrive in extreme conditions. They are extensively found in places like deep ocean bed, salt brine, and also in the volcanic eruption sites. They belong to the Monera kingdom and they are prokaryotes. Archaea are also commonly called as the extremophiles. Archaea are of different shapes. A few are flat and square shaped, while many are in the shapes of spirals, rods, plates, and spheres. The cell wall of Archaea is made of pseudopeptidoglycan. Archaea have been observed to have lipids linked with ether with the branching of aliphatic acid. The reproduction process of Archaea is carried out asexually. The asexual reproduction process is carried out by budding or fragmentation, or even by the binary fission process. There are three types of Archaea identified to date. They are methanogens, thermophiles, and halophiles. Indeed, the Archaea are non-pathogens. Methanogens are widely used for wastewater treatment as it converts carbon dioxide and bacterial waste to methane. Likewise, thermophiles and halophiles have their uses to the human community. The outer cellular layer, called the membrane, plays a vital role in Archaea. They act as the barrier to the cell and the external environment, which makes them thrive very strongly in extreme environmental conditions. Now the question is, what is Archaebacteria? Archaebacteria are single-celled microorganisms living in extreme environments. They form a domain of kingdom Monera. So Archaebacteria are considered to be evolved just after the first life on Earth. Hence, they are called as the ancient bacteria. Archaebacteria are found in hot springs, salt lakes, oceans, and marshlands and soils. They are also found in human skin, oral cavity, and colon as well. Archaebacteria play a vital role in carbon cycle and nitrogen cycle. Their pathogenic or parasitic effect is still not observed. Archaebacteria are metabolically diverse, using a variety of substrates as their energy and carbon sources. Asexual reproduction of Archaebacteria is identified occurring by binary fission, budding, and fragmentation. So individual Archaebacterium is about 0.1 to 15 micrometer in diameter. Different shapes are processed by Archaebacteria like the spheres, rods, plates, and spirals. Some cells are flat, or square-shaped. The cell wall of Archaebacteria is made up of pseudopeptidoglycans. The membrane lipids of Archaebacteria are ether-linked, branched or aliphatic chains containing the D-glycerol phosphates. According to the structure of the cell wall, Archaebacteria are more similar to gram-positive bacteria. So archaebacterial genome contains and consists of a single circular chromosome which exhibits transcription and translation similar to the eukaryotes. Three types of Archaebacteria are found. The first is methanogens, second is halophiles, and third is thermophiles. First I will discuss about methanogens. Methanogens are found in oxygen-free environments like marshes, lake sediments, and digestive tracts of animals producing methane gas. So next is the halophiles that live in water with the high concentrations of salts. So third is the thermophiles that live in hot water environments in acid sulfur springs. So Archaebacteria is shown in figure. This is Archaebacteria as shown into the figure. Next I am going to discuss further about the domain Archaea. So Archaea is a group of primitive prokaryotes that based on their distinct characteristics form a separate domain from bacteria and eukaryotes. The term Archaea is derived from a Greek word, archaeos, which means primitive or ancient, indicating the primitive structure of these organisms. These organisms usually inhabit extreme environments like the deep sea vents, saline waters, hot springs, and even below petroleum deposits. These are mostly anaerobic and live in low oxygen environments. Most of the Archaea cannot be cultured in laboratories and thus have to be identified through culture-independent techniques. Organisms in this domain might share some characteristics with both bacteria and eukaryotes. They have an incipient membraneless nucleus like bacteria but share some genes, metabolic pathways, and enzymes that are also observed in eukaryotes. However, these organisms also have some unique characteristics. The membrane lipids of Archaea contain fatty acid linked to the glycerol molecule by ether bond instead of ester bond as in bacteria and eukaryotes. Because Archaea inhabit many extreme environments, they tend to have distinct metabolic pathways as well as genes that support their survival. Halophilic Archaea have a unique set of genes that limit the extent of osmosis, facilitating their survival. Reproduction in Archaea is asexual by budding, fission, and fragmentation. The usual deviant process of mitosis and meiosis doesn't take place. Most Archaea add the process of biogeochemical cycles for various elements like carbon, nitrogen, and sulfur. Many Archaea are methanogens that utilize anaerobic cellular respiration to produce methane as a byproduct, even though oxygen-generating photosynthesis doesn't occur in these organisms. Some of them, phototrophs, use sunlight as a source of energy. They use numerous sources of energy and display a diverse array of chemical reactions in metabolism. So based on these reactions, they are categorized into nutritional groups. That is either dependent on carbon sources and energy, So one group of Archaea uses sunlight as a source of energy. They are termed as phototrophs. But any of these organisms do not possess oxygen generating photosynthesis. The other forms of Archaea use inorganic compounds as a source of energy, namely ammonia or sulfur. They either include anaerobic methane oxidizers, nitrifiers, and methanogens. This reaction involves two compounds where one compound acts as an electron acceptor and other as an electron donor. The energy that is released during the reaction sites releases ATP. So ATP stands for the adenosine triphosphate. It is one of the same basic processes that can be found in some of the eukaryotic cells. This Archaea domain contains single-celled organisms. Archaea have genes that are similar to both bacteria and eukaryotes. Because they are very similar to bacteria in their appearance, they were originally mistaken for bacteria. Like bacteria, Archaea are prokaryotic organisms and do not have a membrane-bound nucleus. They also lack the internal cell organelles, and many are about the same size as and similar in shape to bacteria. So Archaea reproduce by binary fission, have one circular chromosome, and also use the flagella to move around in their environment as do bacteria. So Archaea differ from bacteria in cell wall composition and differ from both bacteria and eukaryotes in membrane composition and ribosomal RNA type. These differences are substantial enough to warrant that Archaea have a separate domain. So Archaea are extreme organisms that live under some of the most extreme environmental conditions. This includes within hydrothermal vents, acidic springs, and under Arctic ice. So Archaea are divided into three main phyla, and the first phyla is the Cranarchaeota, second is Euryarchaeota, and the third is Corarchaeota. So first I will discuss about the Cranarchaeota. Cranarchaeota include many organisms that are hyperthermophiles and thermoacidophiles. So these Archaea thrive in environments with a great temperature extremes, which are called as hyperthermophiles, and in extremely hot and acidic environments like the thermoacidophiles. So Archaea, known as methanogens, are of the Euryarchaeota phylum. They reproduce methane as a byproduct of metabolism and require an oxygen-free environment. So little is known about the Corarchaeota. Archaea, as few species have been found living in places such as the hot springs, hydrothermal vents, and obsidian pools. So next I am going to discuss about the examples of Archaea. Examples of Archaea. So the first example is Cephalobus. So Cephalobus is a genus of organisms that belong in the domain Archaea and are both acidophilic and thermophilic in nature. They grow at the pH of 2-3 and a temperature of about 80 degrees centigrade. So these are mostly found in the volcanic springs. The proteins found in Cephalobus are particularly important in biotechnology as they are thermostable and also can function at low pH. So these microorganisms are also special because they utilize sulfur as the final electron acceptor during cellular respiration. These are thus dependent on sulfur for the autotrophic or heterotrophic mode of nutrition. So Cephalobus was also used as a model for the study of DNA replication. So multiple sites of origin of replication were identified during studies on these organisms. So some species belonging to this genus are Sulfolobus, Tocodidae, and the Sulfolobus metallicus. So next I'm going to discuss about the methanogens. Methanogens are prokaryotes belonging to the domain Archaea, which are named so because they produce methane as a byproduct during metabolic activities. These are found mostly in wetlands and inside the gastrointestinal tracts of various ruminants and even the human beings. So some methanogens are extremophiles and are found in hot springs and deep sea vents. There are more than 50 species of methanogens known so far, so many of which produce methane through different metabolic pathways. So some methanogens reduce the carbon dioxide in the presence of hydrogen to produce the methane. However, others produce methanol via anaerobic respiration. So, methanogens are widely used in the treatment of wastewater via biocomposition, which is a cost-effective and faster wastewater treatment process. So, some common species of methanogens are Methanosarcina, Barcree, and Methanosarcina acetivorans, and Methanococcus maripludis. So the next question is the very important question, which is what is Eubacteria? Eubacteria are type of bacteria that are single-celled and complex microorganisms that are capable of living and thriving in normal conditions. They are found almost everywhere in the earth, like the soil, water, living and non-living organisms. So Eubacteria also belong to the Monera kingdom, and they are also prokaryotes. Eubacteria are simply known as bacteria as well. So Eubacteria do not have the nucleus and also some membrane organelles. As such, all the processes takes place in the cytoplasm. The cell wall of Eubacteria is made up of peptidoglycan. So Eubacteria has ester links with the fatty acids. The shapes of Eubacteria are spherical, rod, coxy, and spiral, and the bacteria are enclosed in an envelope which protects and regulates the transport of materials. They reproduce sexually as well as asexually, so they are capable of reproducing under the unfavorable conditions too. Eubacteria are pathogens too, meaning they can be harmful and cause sickness in humans. As such, there are non-pathogenic Eubacteria too. There are two types of Eubacteria that exist. So one is the gram-positive Eubacteria and other is gram-negative Eubacteria. Eubacteria follows the glycolysis pathway and Krebs cycle to produce energy. Thymine is present in the transfer RNA of the Eubacteria, whereas introns are absent. Eubacteria are a more complex domain of kingdom Monera. They are found in most of the habitats on Earth, like soil, water, and inside or outside of large organisms. Since Eubacteria do not consist of membrane-bound organelles, almost all the metabolic reactions takes place in the cytoplasm. Some Eubacteria are involved in the nitrogen cycle as well. They also exhibit both parasitic and pathogenic effects on their host organisms. Other than usual asexual reproduction methods, Eubacteria exhibit sexual reproduction methods like conjugation. Individual Eubacterium is about 0.5 to 5 micrometer in diameter. So Eubacteria exhibit a variety of shapes and arrangements. So coxy and bacilli or bacilli are the major shapes. Next is the vibrio. Rods, filaments, and spirochetes are the other shapes of Eubacteria. Membrane lipids of Eubacteria are ester-linked, straight chains of fatty acids containing the L-glycerol phosphates. Eubacteria consist of a single circular chromosome in their cytoplasm. So depending on the cell wall thickness, Eubacteria can be divided into two categories. So first is the gram-positive and second is the gram-negative bacteria. The peptidoglycan layer of gram-positive bacteria binds with the gram stain, giving positive results. The cell wall structure of gram-negative bacteria is also more complex than gram-positive bacterial cell wall and incapable of binding with gram stain. So Eubacteria are shown in figure. This is Eubacteria as shown into the figure. So next I am going to discuss further about the domain bacteria. Domain bacteria. So bacteria are single-celled primitive organisms that form a domain of organisms, diverse in shape, size, texture, and even habitats. So bacteria are prokaryotes that have a membrane, less nucleus, and lack many cell organelles, which make them simple in structure and function. The domain bacteria includes organisms that are found in many different forms of life, from high mountains to inside the body of other organisms. Some bacteria are beneficial that help in various purposes, like antibiotics production, industrial use, and biogeochemical cycles. However, some are pathogenic organisms that results in mild to severe diseases. So bacteria are the smallest living entities in the world and are microscopic. These organisms are observed under a microscope by performing a number of staining techniques. So based on the staining techniques, the bacteria are divided into gram-positive and gram-negative bacteria. Almost all the bacteria have a cell wall made up of the peptidoglycan that protects the bacteria against harmful chemicals. The cytoplasm has a few ribosomes and a membraneless incipient nucleus containing the genetic material. The membrane lipids in bacteria are composed of fatty acids bound to glycerol by ester bonds. Bacteria also have a unique RNA, which is also known as transfer messenger RNA or tmRNA. The genetic material in bacteria is DNA, which is transferred to their offsprings via asexual reproduction. So reproduction takes place through binary fission, budding, and fragmentation, but different methods like the transformation, transduction, and conjugation are available for the transfer of genetic materials. So bacterial DNA, called the nucleoid, are a twisted thread-like mess that flows free. They even possesses a cellular structure that executes a range of circular functions that involves the transfer of energy to the transportation of proteins. Bacteria consist of plasmids, which are a circular piece of DNA. So bacterial cells consist of the inner cell membrane and an outer cell wall, wherein some of the bacteria do not possess a cell wall, such as mycoplasmas. In some cases, the bacteria may consist of a third protective outer layer in a cell, which is called a capsule. So pili cover the surfaces. Bacteria are classified under the bacteria domain, and these organisms are generally feared because some are pathogenic and capable of causing the disease. However, the bacteria are essential to life, as some are part of the human microbiota. These bacteria perform vital functions such as enabling us to properly digest and absorb the nutrients from the foods we eat. So bacteria that live on the skin prevent pathogenic microbes from colonizing the area and also aid in the activation of the immune system. Bacteria are also important for the recycling of nutrients in the global ecosystem as they are primary decomposers. So bacteria have a unique cell wall composition and ribosomal RNA type. They are grouped into 5 main categories. So first is the Proteobacteria. This phylum contains the largest group of bacteria and includes the E. coli, Salmonella, Heliobacter pylori, and Vibrio bacteria. Next is the Cyanobacteria. These bacteria are capable of photosynthesis and they are also known as blue-green algae because of their color. Third is the Firmicutes. These gram-positive bacteria include the Clostridium and Bacillus and Mycoplasmas, the bacteria without cell walls. So, third is the Chlamydia. These parasitic bacteria reproduce inside their host cell. Organisms include the Chlamydia trachomatis, which causes the Chlamydia STD, and next is the Chlamydiophila pneumoniae. Chlamydiophila pneumoniae that causes the pneumonia. So next is spirochetes. These corkscrew shaped bacteria exhibit a unique twisting motion, and examples include the Borrelia burgdorferi. Borrelia burgdorferi causes the Lyme disease. And the next is Treponema pallidum that causes the syphilis. So next I am going to discuss about the examples of bacteria. Examples of bacteria. The first main example of bacteria is the Escherichia coli, that is also known as E. coli. So E. coli is a model organism, microorganism that is used for various research studies. And these organisms are found in multiple environments, and many are found in the lower intestine of human beings and other warm-blooded animals. So most varieties of E. coli are harmless, but few might cause mild to severe diarrhea. Some microbes even produce D, vitamin K, and vitamin B12. So E. coli is a gram-negative and facultative anaerobe that flourishes at the room temperature. So it is a rod-shaped and has short life cycles, which makes it ideal for research studies. And E. coli are non-sporing and have the peritracheous flagella. So next is the lactobacillus. Or lactobacilli. So lactobacillus is a group of the rod-shaped or gram-positive non-spore-forming microorganisms belonging to the family. The term lactobacilli is given to indicate their ability to produce the lactose as a byproduct of glucose metabolism. These organisms are mostly found in milk and milk products. Many varieties of the lactobacillus are used commercially to produce the fermented products of milk and different vegetables. And some commonly used species of this genus are Lactobacillus brevis, and the next is the Lactobacillus Casei or Casei, and next is the lactobacillus plantron. So these organisms are even found within the body of the living beings, like in the intestine and vagina of human beings. Next I am going to discuss about the similarities between the Archaea and Eubacteria. Similarities between the Archaea and Eubacteria. Archaea and Eubacteria are both prokaryotes, meaning that they do not have a nucleus and lack the membrane-bound organelles. They are tiny, single-celled organisms which cannot be seen by the naked human eye, which is called microbes. When we look at them through a microscope, we find that Archaea and bacteria resemble each other in shape and size. They exist as rods, cones, plates, and coils. Both Archaea and bacteria have flagella, thread-like structures that allow the organisms to move, propelling them through their environment. The similarities are that Archaea and Eubacteria are prokaryotes, single-celled organisms that do not have a nucleus or organelles. Size and shape. So both Archaea and Eubacteria are similar in their shape and size. They are both found occurring as rods, coxae, spiral, plates, and coiled. So next I am going to discuss about the difference between Archaea and Eubacteria with table. Differences between Archaea and bacteria. So as we know that Archaea is divided into three phyla. The first phyla is Archaeota, second is the Archaeota, and third is the Archaeota, as shown into the figure. And there are different forms of the bacteria are shown as well. So bacteria are one of the types of biological cells. A large category of prokaryotic microorganisms is bacteria. So bacteria are usually single-celled organisms that live and thrive in different environments. They are present in almost everything: soil, the river, the ocean, even the human feces. The varieties of bacteria are available on this earth. So some are harmful to humans and some are brilliantly useful too. So the categories of bacteria are scientifically based on the nature of their cell walls and shapes. Few are categorized based on the genetic makeup as well. Belonging to the same prokaryotes family based on their genetic makeup, there are two types available. So first one is the Archaea and the other is Eubacteria. So Archaea are called ancient bacteria. Eubacteria are called the true bacteria. So first difference is the size. Archaebacteria, individual Archaebacterium is about 0.1 to 15 micrometer in diameter. So next is Eubacteria. Individual Eubacterium is about 0.5 to 5 micrometer in diameter. So next difference is shape. Archaebacteria are spheres, rods, plates, spiral, flat or square shaped. Eubacteria are coxae and bacilli, vibrio, rods, filaments or spirochetes in shape. Next is the complexity. Archaebacteria are simple in their organization. Eubacteria are more complex than Archaebacteria. Next difference is the habitat. Archaebacteria are found in extreme environments. Eubacteria are found everywhere on earth. So next is the cell wall. Archaebacteria cell wall is composed of pseudopeptidoglycans. Eubacteria cell wall is composed of peptidoglycans with muramic acid. Next character or next difference is the membrane lipids. So Archaebacteria, the membrane lipids of Archaebacteria is ether-linked, branched or aliphatic, chains containing the D-glycerol phosphate. Eubacteria, the membrane lipids of Eubacteria are ester-linked straight chains of fatty acids containing the L-glycerol phosphates. Next difference is RNA polymerase. So Archaebacteria, RNA polymerase of Archaebacteria consists of a complex subunit pattern which is similar to eukaryotic RNA polymerase. Eubacteria, RNA polymerase of Eubacteria consists of a simple subunit pattern. So next difference is the transfer RNA. Archaebacteria, no thymine is present in the TC arm or T-C arm, and it is also known as the T arm. But in some cases, due to the presence of thymine, pseudouridine and cytidine residues, it is also called as the T-C arm of the transfer RNA carrying the methionine. So next is the Eubacteria. Thymine is present in most of the transfer RNA and carrying the N-formyl methionine. So next is the difference in introns. Introns, Archaebacteria, introns are present in Archaebacteria. Eubacteria, introns are absent in Eubacteria. So next is the difference in growth and reproduction. Archaebacteria, asexual reproduction methods like the binary fission, budding and fragmentation are used by Archaebacteria during their reproduction. Eubacteria, other than binary fission, budding and fragmentation, Eubacteria are capable of producing spores in order to remain dormant during unfavorable conditions. And the next difference is the glycolysis and Krebs cycle. Archaebacteria exhibit neither glycolysis nor Krebs cycle. Eubacteria exhibit both the glycolysis and Krebs cycle. So next difference is the types. Archaebacteria are 3 types: methanogens, halophiles and thermophiles. Eubacteria are 2 types: gram-positive and gram-negative. Next I am going to discuss about their examples. So Archaebacteria, the examples are Halobacterium, Lochiarchaeum, Thermoproteus, Pyrobaculum, Thermoplasma and Ferroplasma are the examples of Archaebacteria. Next are the examples of Eubacteria. Eubacteria includes the Mycobacteria and Bacillus, Sporohalobacter, Clostridium and Anaerobacter are the examples of the Eubacteria. An archaea might be very insulted if you mistook it for a bacteria. So however, and vice versa. The differences between archaea and bacteria are profound. As mentioned, the genetic code of ribosomal RNA differs enough to place them in quite different branches of the tree of life, reflecting the different evolutionary paths. This still needs to be confirmed by sequencing the 16 subunit ribosomal RNA of more organisms. Remember how both move via the flagella. Despite this functional similarity and structural similarity, they look similar. They have very different genes encoding them and are comprised of different proteins. Genome sequencing of archaea also reveals genes that resemble eukaryotes more than bacteria. So this is a big difference between archaea and bacteria. Another distinction between these two prokaryotes is the composition of the cell wall. For example, all the bacteria contain the peptidoglycans, which is a molecule and composed of both protein and sugar rings in their cell walls. However, archaea do not have this compound in their cell walls. Cell division in archaea undergoes the distinct processes not found in bacteria. So also bacteria can form spores that lie dormant for years until a proper habitat is found in which they can grow. So Archaea haven't been found to do that. Next is the difference in their cell structure. The general cell structure of Archaea and bacteria are the same, but the composition and organization of some structures differ in Archaea. So similar to bacteria, Archaea do not have the anterior membranes, but both have a cell wall and use the flagella to swim. So Archaea differ in the fact that their cell wall doesn't contain the peptidoglycan and cell membranes uses ether-linked lipids as opposed to ester-linked lipids in bacteria. So next character is the flagella. Archaea flagella evolved from bacterial type 4 pili, while the bacterial flagella evolved from the type 3 secretion system. So the bacterial flagellum is like a stalk, which is hollow and is assembled by the subunits that are free to move up the central pore adding on the tip of flagella, while in Archaea, flagella subunits are added onto the base. So next character is the reproduction and growth. Archaea reproduce asexually by the process of bindiffission, budding and fragmentation. Eubacteria reproduce asexually through bindiffission, budding, fragmentation, but Eubacteria have the unique ability to form spores to remain dormant for years and over years, a trait that is not exhibited by Archaea. So bacteria growth follows in 3 phases. The lag phase, when cells adapt to new environment, and log phase, marking the exponential growth, and stationary phase when nutrients get depleted. And the next character and the next difference is habitat. Archaea can survive in extreme and harsh environments like the hot springs, salt lakes, marshlands, oceans, gut of the ruminants and humans. Eubacteria are ubiquitous and are found in soil, hot springs, radioactive wastewater, earth's crust, organic matter, bodies of the plants and animals, etc. So next I am going to discuss the Archaea versus Eubacteria. Archaea versus Eubacteria. The difference between the Archaea and Eubacteria is that Archaea is a single-celled bacterium that thrives in extreme conditions, while the Eubacteria live and thrive in normal conditions. Archaea is the oldest bacteria ever known while the earth was formed. Both Archaea and Eubacteria belong to the kingdom which is called Monera. So first I will discuss the Archaea, then I will discuss the Eubacteria. So Archaea versus Eubacteria. Archaea. Archaebacteria are simple in their organization. Eubacteria are more complex than Archaebacteria. Archaebacteria or Archaea found in extreme environments. Eubacteria found everywhere on earth. Archaea, sphere, rod, plate, spiral, flat or scale-shaped. Eubacteria coxy, bacillus, vibrio, rods, filaments or spirochetes in shape. Archaea, cell wall is composed of pseudopeptidoglycans. Eubacteria, cell wall is composed of peptidoglycans with muramic acid. Archaea, introns are present. Eubacteria, introns are absent. Archaea, exhibit neither glycolysis nor Krebs cycle. Eubacteria, exhibit both glycolysis and Krebs cycle. Archaea has 3 types. Eubacteria has 2 types. Archaea includes methanogens, halophiles and thermophiles. Eubacteria includes the gram-positive and gram-negative bacteria. Archaea, asexual reproduction methods like the binary. Eubacteria, capable of producing spores in order to remain. Archaea includes fission, budding and fragmentation are used, and Eubacteria remains dormant during the unfavorable conditions. Archaea, RNA polymerase consists of a complex subunit pattern. Eubacteria, RNA polymerase consists of a simple subunit pattern. Archaea. Membrane lipids are ether-linked branched aliphatic chains containing the D-glycerol phosphate. Eubacteria. Membrane lipids are ester-linked straight chains of fatty acids containing the L-glycerol phosphates. Archaea. Examples include Halobacterium, Lochiarchaeum, Thermoproteus, Pyrobaculum, Thermoplasma, and Ferroplasma. Eubacteria. The examples include Mycobacterium or Microbacteria, Bacillus, Sporohalobacter, Clostridium, and Anaerobacter. So next I am going to discuss about the comparison table between the Archaea and Eubacteria in tabular form. So first I will discuss the parameter of comparison, then I will discuss Archaea, and in the last I will discuss Eubacteria. So first is the meaning and definition. Archaea. Archaea are a type of bacteria that are single-celled and simple microorganisms that are capable of living and thriving in extreme conditions. They belong to the Monera kingdom. Eubacteria are a type of bacteria that are single-celled and complex microorganisms that are capable of living and thriving in normal conditions. They also belong to the Monera kingdom. So next is the places of existence. Archaea. Archaea are widely found in very unusual environments like the ocean depth, volcanic sites, and salt brine. Eubacteria are found almost everywhere. It is found in all the living and non-living organisms like the soil and water, etc. So next is the reproduction. Archaea follows asexual reproduction. This usually happens by the binary fission, fragmentation, or at times by budding process too. Eubacteria produces spores. This allows them to live in not so favorable conditions also. They reproduce sexually as well as asexually. Cell wall. The cell wall is pseudopeptidoglycan in Archaea. Eubacteria. The cell wall is made up of peptidoglycan with the muramic acid or sometimes with the lipopolysaccharide. Next is impact factor. Impact factor. Archaea are non-pathogens, meaning they are not harmful and never causes the diseases in humans. Eubacteria are pathogens, meaning they may cause diseases in humans. So next I am going to discuss the comparison chart. First I will discuss the differences of Archaea, and then I will discuss the differences of Eubacteria. First difference is in introduction. Archaea constitute a domain or kingdom of single-celled microorganisms. These microbes are prokaryotes, meaning that they have no cell nucleus or any other membrane-bound organelles in their cells. Next is Eubacteria. Bacteria constitute a large domain of prokaryotic microorganisms, typically a few micrometers in length. So bacteria have a number of shapes, ranging from spheres to rods and spirals. Next difference is in cell wall. Archaea, pseudopeptidoglycan, Eubacteria, peptidoglycan and lipopolysaccharide. Next difference is in habitat. Extreme and harsh environments like the hot springs, salt lakes, marshlands, oceans, gut of ruminants and humans. Eubacteria are ubiquitous and are found in soil, hot springs, radioactive wastewater, Earth's crust, organic matter, bodies of plants and animals, etc. And the next difference is in growth and reproduction. Archaea reproduces sexually by the process of binary fission, budding and fragmentation. Eubacteria reproduces sexually through binary fission, budding, fragmentation. But Eubacteria have the unique ability to form the spores to remain dormant for over years, a trait that is not exhibited by Archaea. So next I am going to discuss the main differences between Archaea and Eubacteria. Main differences between Archaea and Eubacteria. Archaea and Eubacteria belong to the same Monera kingdom. However, they differ in many aspects. The main difference between Archaea and Eubacteria is the Archaea live and thrive in extreme conditions, whereas Eubacteria can be found almost everywhere. Archaea reproduces asexually by binary fission, fragmentation, and budding process, while Eubacteria reproduces both asexually as well as sexually. Archaea are completely non-pathogens, while Eubacteria have both pathogenic and non-pathogenic bacteria. The cell wall of Archaea is made up of pseudopeptidoglycan, as they also have eta bonds with the aliphatic acid, while Eubacteria have a lipid membrane ester links with fatty acids. So Archaea do not follow the glycolysis or Krebs cycle for metabolic activities, but follow a similar pathway, whereas Eubacteria follows the glycolysis and Krebs cycle to break down the glucose and produce energy. Archaebacteria and Eubacteria are two domains of the kingdom Monera, which contains the least organized unicellular prokaryotic microorganisms on Earth. Both Archaebacteria and Eubacteria are single-celled microorganisms which are usually called as prokaryotes. The main difference between Archaebacteria and Eubacteria is that the Archaebacteria are usually found in extreme environmental conditions, whereas the Eubacteria are found everywhere on Earth. So next I am going to discuss about the key differences Archaea versus Eubacteria. Basis for the comparison, first I will discuss the Archaea and then I will discuss the bacteria. First is definition. Archaea is a group of primitive prokaryotes that based on their distinct characteristics form a separate domain from bacteria and eukaryotes. So bacteria are single-celled primitive organisms that form a domain of organisms diverse in shape, size, texture and even habitats. Next is the character of the pathogenicity. Archaea are non-pathogenic. Bacteria might be pathogenic or non-pathogenic. Next is habitat. So most Archaea are extremophiles and are found in extreme environments like the deep sea, mountains, hot springs and salt brine, etc. Bacteria reside in different habitats ranging from soil, water to inside living and non-living organisms. Next is reproduction. Archaea reproduce by fission, budding and fragmentation, and sporulation doesn't occur in Archaea. So some bacteria are capable of forming the spores that help them to survive in extreme conditions for a particular period of time. Next is the cell wall. The Archaea cell wall is made up of pseudopeptidoglycan and lack the D amino acids and N-acetylmuramic acid. The bacterial cell wall is made up of peptidoglycan consisting of N-acetylmuramic acid and D amino acids. Next character is the membrane lipid. The fatty acids and membrane lipids of Archaea are bound to the glycerol by the eta bonds. The fatty acids and membrane lipids of bacteria are bound to glycerol by ester bonds. So next character is the glucose oxidation. Archaea do not use the glycolysis or Krebs cycle for glucose oxidation, but follow the metabolic pathways similar to these. Bacteria, glycolysis and Krebs cycle are important metabolic pathways in bacteria for glucose oxidation. So next character is the photosynthesis. Archaea do not perform oxygen generating photosynthesis but are phototrophs that use the sunlight as a source of energy. Many bacteria containing the photosynthetic pigments that can perform the photosynthesis to prepare their own food. And the next character is the types. Archaea are divided into different groups like methanogens, thermophiles and halophiles on the basis of their characteristics. Bacteria are divided into gram-negative and gram-positive based on their response to gram staining. And the next character is flagella. So the archaea flagella, also termed as the archaea, are synthesized by adding the subunits at the base, and bacterial flagella are hollow and assembled by adding the subunits moving from the central pore towards the tip of flagella. So next character is the transfer RNA. The thymine is absent in the transfer RNA of archaea. Thymine is present in the transfer RNA of bacteria. Next character is the tmRNA. tmRNA stands for the transfer messenger RNA that is found in archaea, and transfer messenger RNA is also found in bacteria. So next character is the chromosomes. Introns are present in the chromosomes of archaea, introns are absent in the chromosomes of bacteria. So next character is the RNA polymerase. RNA polymerase in the archaea is complex with more than 8 polypeptides. They might have multiple RNA polymerases. Bacteria RNA polymerase is simple with the 4 polypeptides. Examples of the archaea are Thermosphaera aggregans, and next is the Staphylococcus marinus. Next is the Sulfolobus tocodi, and the examples of the bacteria are Pseudomonas aeruginosa, and next example is the Bacillus subtilis. Next example is the Staphylococcus aureus, and the next example is Salmonella typhi or Salmonella typhi. So next I am going to discuss about the Archaea, bacteria and eukarya. Archaea, bacteria and eukarya, all the living things can be classified into a place on the tree of life. So this phylogenetic tree has 3 major branches which are called Archaea, bacteria and eukarya. A phylogenetic tree traces the evolutionary history of organisms and indicates the common ancestors. We already see a major difference between archaea and bacteria from this classification. They have a different evolutionary history as they occupy very different places on the tree of life. So how was this tree of life composed? The sequence of 16 subunit ribosomal ribonucleic acid or 16 subunit ribosomal RNA, a fundamental unit of ribosomes, was compared across organisms. This showed that the underlying genetic code for a component of ribosome differed greatly between the archaea, bacteria and eukarya. Thus they formed the 3 distinct branches of the tree of life, and there was a time when archaea were not understood to be different from bacteria, and in fact were. Erroneously named Archaebacteria, and the tree of life was drawn wrong. But they are definitely not bacteria. Even though archaea and bacteria share some similarities, archaea deserve their own branch on the tree of life. So this is the phylogenetic tree of life. So there are 3 kingdoms: bacteria, archaea and eukaryota. So the bacteria includes green filamentous bacteria, gram-positives, spirochetes, proteobacteria, cyanobacteria, planktomyces, bacteroides, cytophaga, thermotoga and equifax. Archaea includes halophiles, methanosarcina, methanobacterium, methanococcus, T-celler, thermoproteus and pyrodicticum. On the other hand, eukaryota includes the entamoeba, slime molds, animals, fungi, plants, ciliates, flagellates, trichomonads, microsporidia and diplomonads. So next I am going to discuss about the history of phylogenetic classification. So history of phylogenetic classification: until the middle of the 20th century, biologists classified all the living things as either a plant or an animal, but this system failed to accommodate the fungi, protists and bacteria. So by the 1970s, the classification system evolved to what was known as 5 kingdoms. That includes the prokaryotes, bacteria and eukaryotes like the plants, animals, fungi and protists. Eukaryotes are characterized by the presence of nuclei, cytoskeletons, and internal membranes in their cells. In the late 1970s, the Dr. Carl Woese and his colleagues at the University of the Illinois identified a group of microorganisms whose genetic makeup was vastly different from other bacteria. So they divided the prokaryotic life into what they called the Archaebacteria and Eubacteria. However, they later concluded that Archaebacteria were sufficiently different as to not to be bacteria at all. So the groups were renamed to Archaea and Bacteria. So Archaea is divided into three phylums, which are Archaeota, Archaeota, and Archaeota. On the other hand, the bacteria is further divided into the Vibrio, Spirillum, Bacillus, and Coccals. And the Eukaryota is further divided into the plants. Plants are divided into the vascular, non-vascular, with seed or seedless, are divided further into gymnosperms and angiosperms. On the other hand, the animal kingdom is further divided into the cnidarians and bilateral. Bilateral is further divided into the crustaceans and arthropods. Arthropods are further divided into the arachnids and myriapods and insects. On the other hand, bilateral is further divided into the vertebrates, and vertebrates are divided into bony fishes and divided into tetrapods, and tetrapods further divided into amphibians and are further divided into the amniotes, and amniotes are further divided into the mammals. So mammals are further divided into the placentals, and mammals are further divided into marsupials, and mammals are further divided into monotremes. On the other hand, the amniotes are further classified or divided into birds and reptiles. So birds and reptiles are further divided into the turtles, lizards, and snakes. And the vertebrates may be further divided into cartilaginous fish, and bilateral is further divided into the mollusks. On the other hand, fungi kingdom is further divided into basidiomycetes, zygomycetes or ascomycetes or ascomycetes, and the next is the protista. These all are shown into the figure. So next I am going to discuss about the conclusion. Archaea and Eubacteria are single-celled microorganisms that show some similarities morphologically but differ in many other properties. So being in the prokaryotes family, they lack the cell membrane and nucleus, and the need for both types of bacteria is of immense help to the humankind. Archaea has a lot of benefits to mankind, and so does Eubacteria. Methanogens are widely used in the production of biogas and also for the sewage water treatment. Eubacteria supports the digestion of food and also helps preparing the curd and yogurt. The Eubacteria help the people from infected with the certain sicknesses too. So Archaebacteria and Eubacteria form a prime part of the earth, without which mankind cannot survive. So Archaebacteria and Eubacteria and Cyanobacteria are the three domains of the kingdom Monera. Archaebacteria are called the ancient bacteria, whereas Eubacteria are called the true bacteria. Eubacteria are usually found in soil water, living in and on of large organisms. Eubacteria are divided into two groups, known as gram-positive and gram-negative bacteria. So Archaebacteria are found in salt brines, ocean depths, and hot springs. They have evolved just after the evolution of first life on Earth. The three types of Archaebacteria are found, which are methanogens, halophiles, and thermoacidophiles. The main difference between Archaebacteria and Eubacteria is their habitats in the environment. If you like my video, please subscribe to my YouTube channel and press the bell icon as well for further notifications and updates. Thanks for watching. Thank you so much for your time and appreciation.